All right. Good morning, everybody. Thanks for taking your time on Saturday to learn about our latest research project with Dr. George in Northwestern. This project in this presentation is intended to be like a layman explanation to those of us who aren't research professionals and professional scientists. The project is a joint venture. It's a collaboration between the HC Foundation and CureHC and Hope for Annabelle and the For Henry group. It's another collaborative project. And the goal of today is to give us all a pretty good understanding about what we've been funding recently, and to give us all that kind of elevator spiel that we can pass on to the next group, or you're talking to somebody and you want to raise some funds, and just to give that couple minute explanation to someone else. So big thanks to Dr. George for bringing this information and making it accessible to all of us. And I appreciate it. This is a little bit of a preview to his presentation in Edinburgh, but it is going to be a little different for us. So I very much appreciate the extra effort, Dr. George.
Great. Thank you very much, Josh. And thank you everyone who's joined this morning. I appreciate the enthusiasm and willingness to give up part of your Saturday mornings. My plan today is to tell you about some ongoing research in our laboratory at Northwestern that seeks to determine if antisense oligonucleotide therapy is right for AHC. And I'll explain what that jargon means as I go through this morning. But let me begin by just saying to you that classical pharmacology involved small organic molecules that we consider normally drugs. But in the last decade or so, there's been an emergence of less traditional medicines. Monoclonal antibodies is a good example for treating inflammatory conditions and cancer and other things. But recently, there's been an emergence of different classes of biological molecules that involve nucleic acids. And those are either RNA or DNA. And here's a list of 15 approved FDA-approved drugs that are, in fact, nucleic acid drugs. And these all fall into this category of antisense oligonucleotides. I'm not going to go through the list. I just highlighted one of them, which is the name of the drug is nusinersen. It's sold under the brand name of Spinraza. And this was developed for a rare, often fatal neurological disease affecting infants and young children called spinal muscular atrophy. And this disease is now effectively treated by using this drug, nusinersen. But importantly, unlike conventional drugs that can be made into pill form and swallowed, antisense oligonucleotides, as illustrated here for nusinersen, has to be given differently. For treating a brain disease, or in the case of spinal muscular atrophy, a disease of the spinal cord, you have to inject it directly into the space surrounding the spinal cord. That is called a lumbar puncture or spinal tap, as illustrated in this cartoon. And then the drug diffuses through the cerebral spinal fluid and can reach the brain. Now for Spinraza, this is the dosing regimen. It has to be given initially multiple doses over the first month, and then it's dosed every 4 months thereafter for life. That's the important thing, for life. Now, there are other FDA-approved antisense oligonucleotides that were developed not for a large population of patients, but rather for one patient. And here is a breakthrough report from 2019 in the New England Journal of Medicine. And this was an effort, a huge team effort led by Timothy Yu at Boston Children's Hospital, which developed a customized therapy for one patient, a young girl with a rare genetic disease called Baffin's disease. And to skip through sort of the details of that, I just want to highlight the fact that the reason this paper has received a lot of attention was because of the extraordinary and accelerated time frame for performing the basic experiments and bringing this to the point where the FDA gave it approval for treating one individual. The timeline is shown here. The patient came into the hospital in 2016. A diagnosis was made. They had a couple of months went by before they had genetic testing. Once they had the genetic test results, there was a clear pathway to potentially correct the mutation, which generated a toxic product in the nervous system. And they used antisense oligonucleotides to get rid of that toxic product. The challenge, of course, was nothing like this had ever been done on a single individual. And they put a lot of time, a lot of money to push this in an accelerated way. After 18 months, they actually got approval to put the first dose in the child. The child was having daily seizures on the order of 20 to 30 seizures a day, and treatment with this new antisense oligonucleotide, which was named Millicin, a derivative of the child's name, was effective and reduced her seizure burden over the course of a couple of months. Now, it wasn't life-saving in the end, but it did demonstrate for many people the idea of using a personalized approach to develop a single therapeutic compound for an individual. Now, I'm talking about an approach not to develop a drug for one person in this case, but to try to develop a new concept for a drug that could affect or benefit many people with alternating hemiplegia of childhood. But this is out there, and this has gotten many people in the rare disease community very excited about antisense oligonucleotides. So they are all the rage, very popular. You hear about them all the time when you attend meetings where rare disease therapeutics is discussed. It may or may not work for alternating hemiplegia. Let's dive into this. Before we get into the nitty-gritty, I have to back up and train all of you in the science of molecular biology. This is something that I think about very routinely, and I know that it's not something everybody understands. So here's the core concept of genetics and what's called molecular biology. And that is that DNA, pointer here, DNA carries the instruction set for a cell to manufacture every protein it needs for function and survival. That DNA code is read first by converting it to what is called messenger RNA, simply here as RNA, which is another nucleic acid. Then the RNA is read by the machinery in the cell that generates proteins. So this is the central dogma. DNA is transcribed into a message called RNA, and then the RNA message is read and translated into protein by the cell. Now, DNA, we widely understand, is a unique molecule. The nature of the molecule was revealed back in the 1950s by these two scientists, Watson and Crick, and they won the Nobel Prize for this achievement. And they, modeling here, are showing off their structure of what is now known as the double helix. The double helix is simply a chemical structure that is built upon a curious interaction between four different chemicals shown on the left. These are known as nucleobases or bases. We just call them bases. There's thymine, cytosine, adenine, guanine, and you can actually abbreviate these TCAG for short. Now, they're just part of the machinery, and they're color-coded here in the DNA helix. This is basically two ribbons of molecules that interact with one another by chemical interactions between the bases. And those chemical interactions are not random. They're very specific. Thymine, for example, interacts with adenine, and cytosine interacts with guanine. Hence, you see the color coding here. Yellow and red are always paired. Green and blue are always paired. That's what keeps the double helix together. That's like the teeth of a zipper, if you think of it that way. Now, RNA is very similar, except instead of two strands tied together with these interactions, it's one strand. And the composition is very similar to DNA with one exception. It has guanine, adenine, cytosine. But instead of thymine, it has a slightly different molecule, uracil. And so when we think of DNA, we think of the bases as TCAG. When we think of RNA, it's UCAG. Very similar. Those structures are tied together chemically in a reversible way. I won't get into the details, but it's the bases that contact and talk to each other. Each base is tied to a sugar molecule and also a phosphate group to link them together. And so this is the ribbon part of DNA or RNA. And again, that base pairing abbreviated as C and G and T and A or U and A for RNA. And these are base pairing phenomenon that chemically are often referred to as Watson-Crick base pairing. You may hear that sometimes. So again, this is the structure of DNA. On the right is simply the helical representation of that. And then on the left is simply, if we take away that helical appearance, we can again see the base pairing adhering or gluing these two strands together always has G and C paired and T and A paired. Now, RNA being single, a single strand, you would think it's always just a linear molecule, but it's not. Actually, it turns out RNA can also undergo these same kind of base pairing reactions and interactions. The difference is it just folds on itself, and it can make stretches that look a lot like the double helix. And then other sections that it's not, it's single. And so this is important to know because RNA is a complicated molecule in how it folds. And sometimes we may need to know more about how it's folding so that we can pick a region to target with an antisense oligonucleotide. Another important principle is that RNA can actually bind to DNA. So DNA is two strands that interact with each other through this so-called Watson-Crick base pairing. RNA is single-stranded. It can either fold on itself or possibly, if you can split DNA into its separate strands, RNA can sneak in and bind to it. So you can actually make an RNA DNA sandwich, if you will. That is one of the principles behind the use of antisense oligonucleotides. And the reason for that is this unique combination of RNA and DNA signals to the cell in a particular way that this should be degraded. So here is the fundamental principle of antisense oligonucleotides. Well, first of all, let me break down these words for you. Oligonucleotide is simply a short stretch of DNA or RNA, or possibly a mixture of RNA and DNA. Oligonucleotide simply means it's a small number of nucleotides. There's no set number. It could be 10, it could be 20, it could be 50. It's just a term that we use to classify the broad chemical nature of short stretches of synthetic DNA or RNA or a mixture of the two. Antisense refers to the fact that the sequence of the bases, as an example here, A, C, T, T, G, T, A, A, C, A. The antisense is just the reverse strand that is set up to bind to it. So in this side, there's A, and this side, there's T. There's a C, and here's a G. And then there's a polarity to it. 5', 3' is illustrated here. So the antisense simply references the fact that we want a sequence that will be interacting with another sequence, the target sequence. And it simply is the bases are arranged in that sequence so that they form this zipper-like structure. So the fundamental concept here is we can design and synthesize an oligonucleotide that is antisense to a messenger RNA molecule. And so what's shown here again is the central concept of genetics and molecular biology. DNA is codes for proteins, but it has to first be converted to messenger RNA, which is then the messenger RNA is converted to read and allows the cell to make the protein. An antisense oligonucleotide will seek out a stretch of the mRNA sequence, bind to it, and create this unique double-stranded zone where you have RNA on one side and DNA on the other side. And this RNA DNA complex is recognized by an enzyme in the cell called ribonuclease H. Back up. And ribonuclease H cuts it and cleaves it. And ultimately, this prevents that messenger RNA from making the protein. So this is a strategy. An antisense oligonucleotide is a strategy to target a very specific sequence in messenger RNA and target it and mark it for destruction in the cell so that you can interrupt this central dogma of molecular biology and not make a particular protein. Now, therapeutically, DNA only doesn't work very well. So there's been a lot of interest in using a particular design of antisense oligonucleotides in which a stretch of DNA, which is going to do all the work of engaging RNase H, is in the core. And then flanking it are chemically modified RNA bases. So it's RNA on one side, a core of DNA, followed by some more flanking RNA. And this, I don't know who came up with this name, but this is referred to as a gapmer. I guess the gap is the DNA part flanked by RNA, but gapmer is the common method to design antisense oligonucleotides to engage RNase H, which again is this enzyme in the cell that will destroy a messenger RNA. Now my animation's a little out of order here, or my brain is out of order probably. But I want to point out a couple of things. One is when designing an antisense oligonucleotide to target, say, a mutation in a gene like ATP1A3, we have to recognize that you are dealing with two conditions. Either you're making it specific for a mutation, which we would call allele specific, or it's targeting the gene independent of the mutation, in which case it would be allele non-specific. For the case of AHC, what I'll be showing you are all examples where we're trying to achieve allele specific targeting. We don't want to target the normal sequence of mRNA for ATP1A3. We want to only target the mutant sequence. Because at the end of the day, we only perhaps want to destroy the mutant sequence. And here is where this jargon of knockdown comes in. When RNase H destroys this double-stranded segment of messenger RNA, the entire messenger RNA is degraded by the cell. and you don't make the protein. That means if you measured the levels of messenger RNA in the cell, they would be lower. You've destroyed it. Same thing, if you measured the level of protein in the cell, it would also be lower. And so we have this term knocks down or knockdown. We're lowering or reducing or knocking down the level of either messenger RNA or protein or both, depending on what you're measuring. So knockdown is simply a term used to say we're reducing the level of ATP1A3 mRNA or protein. So going forward, now I'm going to really go away from the fundamentals. Hopefully I explained some of that well enough for some of you. The three things I want to talk about next, or for the rest of the time, are our attempts to answer these three questions. Well, the first question, actually there's four questions. The first question is, will antisense oligonucleotides, abbreviated ASO, will they work for AHC? It's not clear they will work. We think there's a path to figure out if they will work or not. But first of all, we have to determine whether eliminating the mutant copy of ATP1A3 would be beneficial. Would it help if we got rid of that? Second, if we are designing this to get rid of the mutant RNA for the mutation for the ATP1A3 mutation, can we make an ASO that only affects the mutation and doesn't attack the healthy copy? And then finally, kind of conceptually, if this works, will there need to be a different drug for every single ATP1A3 mutation? Or is there a way around that? Those are our three quests in our current research study that's been generously funded now by a consortium of the ATP1A3 and AHC foundations, which we greatly appreciate. So let's talk about the first question. Would eliminating the mutant allele be beneficial? And for that, to answer that, we took a step back and asked the common question, which is, what's the basic molecular mechanism of AHC? What's causing this disease? We know the gene. We know there are mutations in that gene. And so far, I think the evidence is strongly supportive of the idea that the mutations in ATP1A3 disable ATP1A3. They cause loss of function of ATP1A3. ATP1A3 is an ion pump present in neurons, and these mutations prevent the pump from doing its job. That, I think, is very well established. What is a little less clear is we have two copies of ATP1A3. When one of them has a mutation in it that causes loss of function, is the disease a manifestation of having one and only one healthy copy of the gene? Or does that mutation interrupt the function of the healthy copy in some indirect or direct way? And to put fancy terms on those two concepts, haploinsufficiency is a disease mechanism whereby the disease occurs because one healthy copy isn't enough for health, and that results in a disease. By contrast, dominant negative is the term we use for situations where there's one mutation, one mutant copy, and one healthy copy of a gene. And for some reason, that mutant copy suppresses the activity of the healthy copy. And so getting rid of the healthy copy, or getting rid of the mutant copy, may be beneficial. Not beneficial in the setting of haploinsufficiency. It doesn't probably matter as much there. So in AHC, the evidence really is favoring a dominant negative effect. That evidence is listed here. We don't see overt loss of function mutations in the gene. What I mean by that is we don't see mutations that truncate the gene or truncate the protein. that we can predict easily are complete loss of function that would likely not interact with the healthy copy. We just don't see those. So that would be an argument against haploinsufficiency. Another argument against haploinsufficiency is the fact that people have made mice where they're missing one copy of ATP1A3, officially called heterozygous knockout mice. They do not develop AHC. There are some subtle neurodevelopmental abnormalities, but they do not have the severe AHC that's been observed in mice that have mutations placed in one of the copies. And then lastly, there is one paper by Steve Petro where they demonstrated in a frog oocyte, an expression system that's used for studying ATP1A3 function, they demonstrated dominant negative effects of the three most common AHC mutations. So the evidence is favoring dominant negative effect. Is it airtight? Probably not. Is it close? It's close enough for us to think that we should test whether or not an antisense oligonucleotide strategy would work in this condition. So that's one of our first goals. So just to explain this even further, just so everyone is on the same page, here's what I mean by haploinsufficiency. So you have two copies of ATP1A3. One is the healthy copy in blue, and the other is the mutant copy in red with a little star. Haploinsufficiency simply means the disease occurs because one copy of the gene is not enough for health. By contrast, a dominant negative mechanism, you start out with the same pairing of a healthy copy and a mutant copy, but for some reason, the mutant copy poisons the healthy copy. Probably at the protein level, but there are many possibilities. If this is the case in AHC, then an antisense oligonucleotide approach diminishing the mutant would leave the healthy copy alone, and we would have health and not the disease. So that's the concept. If it is dominant negative and we can direct an ASO to only knock down the mutant copy, we might have a therapy for AHC. So we have been studying AHC for a few years using human neuron models of the disease. And these models are derived by a technology known as induced pluripotent stem cells, or iPS cells. And this is a technology that's been around now for several years, and it's really revolutionized biology and research in the biomedical fields. For years, in collaboration initially with Kevin S. at Vanderbilt, we collected many patient samples, blood samples, some skin samples. We also had some skin samples from Kathy Swoboda, then at Utah, now in Boston. And so we now have 17 or 18 different iPS cell lines with these mutations representing AHC. And what we can do with that is these particular cells can be made into neurons. And here's just some photos from the lab where we've made two types of neurons. We've made the so-called excitatory neurons and inhibitory neurons. And these can be used for all kinds of research in the laboratory, from electrophysiology to biochemistry to molecular biology. And this has been our platform now for testing whether or not an ASO strategy would be beneficial for AHC. So to start out, we've actually recently performed a key experiment, which is, what if we just simulate what would happen if you converted an AHC neuron to one where the mutant copy is gone and you only have the healthy copy. So to do that, this is, of course, not a therapeutic approach. This is an engineering approach in the lab. We used what is called CRISPR-Cas9, or just simply CRISPR, which is the term used for a gene editing technology and this is just an illustration of it. This is essentially a targeting strategy that can allow these active molecules, Cas9 in particular, to hone in on a specific stretch of DNA in a cell. And they can then cut it. It cuts it, and then the cell has repair mechanisms to repair that cut, but it may repair it in a way that's not functional. It'll either delete something or insert something, and at the end of the day, it disrupts the gene. So we applied this technology, and in a cell line carrying the D801N mutation, we were able to delete that mutant copy of the gene. And then we had cell lines that only had the healthy copy. And we wanted to know what properties they have when we do that. So we looked at two things. We looked at the mRNA for ATP1A3, and we looked at the protein level for ATP1A3. Did we see knockdown? So a curious thing we observed. So first of all, here is a time course of the mRNA. Shown in red is the original cell line with the mutation and the healthy copy. So this is a cell line from someone with D801N related AHC. The RNA level, which we've measured in a way that we normalize to another gene to make sure that the measurement's accurate, the mRNA in the mutant cell line doesn't change much over the course of 35 days. However, this cell line that we made, which I call the het knockout, heterozygous knockout, het KO, where there's only the healthy copy, the mRNA rises over the course of 35 days. It's not static. We get more. The cell makes more. Also, at the protein level, now we're measuring protein, and we have to normalize it to a different marker. At day 14, which is early, the neurons have in the heterozygous knockout, they have, as we predict, relative to the original cell line, which is the mutation plus the healthy copy, they have half of the amount of protein, which is what we would expect. But over the course of the next 3 or 4 weeks, protein levels change. The red dots here show you the protein level in the original cell line, which is the mutation cell line with their mutant and the healthy copy. That doesn't change. Interestingly, the protein level in the heterozygous knockout level climbs and eventually reaches a level that's equivalent to where the other line is. So we interpret this as the fact that maybe human neurons have the ability to compensate for losing a copy of ATP1A3. They start out low, but something happens and they compensate and they make more from that one copy of the gene. So that bodes well. If we can design an ASO to do the same thing, can that work? So that's where we're pivoting now. So can an ASO be designed now so specifically that it only attacks the mutation? So again, we frame this around this common mutation, D801N. We have other projects using approaches for E815K as well. But we adopted the gapmer design strategy. We designed and synthesized a series of antisense oligonucleotides that are spanning the mutation. Here is the D801N nucleotide sequence showing the mutation in black here, and that should be a G. It's an A instead. And here are our gapmer designs, just by cartoon, that basically span this region and slowly walk over this region one base pair at a time. So we make a panel of ASOs all through this region because we don't know which sequence specifically is going to work best. Also importantly, the standard of the field right now is to not use native RNA and DNA sequences, but rather to make chemical modifications, particularly if the RNA flanking the DNA core. And those RNA bases are made where the sugar backbone is modified with some extra chemistry. In particular, it has a methoxyethyl group stuck to one of the oxygens off this sugar molecule. I know I'm getting into the chemical weeds here, but that enhances two things. It improves stability, so it won't be broken down by enzymes in the body, but it also affects the stability of how it interacts with DNA. The other thing is, instead of phosphate with oxygens, there's a way of putting sulfur in there, and that also improves the stability. So we have this modification as well. So these are chemically modified bases that are engineered to work well as an antisense oligonucleotide. Now, the experiments that we've been doing, we grow neurons in the lab. In this case, we grow either the excitatory or the inhibitory neurons from a cell line, an iPS cell line that has the D801N mutation. We culture them for several weeks, several days, probably up to 6 or 7 weeks. Then we introduce the ASO into the cultures for 6 or 7 days. And then at the end of the experiment, we isolate RNA and we measure whether or not the RNA for the healthy copy or the mutant copy has changed. So first of all, appreciate the fact that these are long experiments. This is 8 to 9 weeks of work for one experiment. And so we try to do things in parallel, but it does take time. And this is one of the frustrating things about this platform, is it does take time. Well, anyway, here's some of our first work where we designed a series of ASOs against D801N. We transfected them, in this case, into the inhibitory neuron class. And then we separately measured ATP1A3 healthy copy or mutant copy at the mRNA level. This is mRNA. And then we express them as a percentage. So if we did not treat with an ASO, everything is 100%. Wild type and the mutant, they're there and in equal amounts. And now what we're looking for is an ASO that knocks down the mutant message, but doesn't do much to the healthy copy. So here's the results. Most of these ASOs in this first batch didn't select very carefully between the mutant and the healthy copy. They just knocked them all down. There were a few that were a little bit different. If we look at the measurements actually for the two favorite ones, number 2 and number 5, number 2 knocked everything down a lot. But number 5 knocked the wild type down, which is the healthy copy, by 50%, and the mutant by left only 28%. So we thought there was a little difference there. We did the same thing in the excitatory neurons. This is a different batch of neurons. And basically we see a similar arrangement where these ASOs aren't very selective. There's some selectivity, but not very much. So we get good knockdown of the mutant, but we're also knocking down the healthy copy as well. We did a dose response curve, hoping that maybe at a lower concentration, we would get better selectivity than a higher concentration. It turns out we didn't. We see the same preference for both copies from these. There is some selectivity. It's just not probably enough. So we've had a few rounds of trying to improve the selectivity. Here is one in which we changed the overall length of this gapmer design to shorten the RNA sides and the DNA sides. And here we can see a difference between mutant and wild type. It's just we don't preserve the wild type well enough. While we would like to get this level of knockdown for the mutant, but preserve the wild type. We haven't seen that yet. We tried something else recently where we introduced some mismatched bases where we would disable or disrupt the interaction with the healthy copy, but preserve the interaction with the mutant copy. And I just noticed the colors are switched here. So on the right, the mutant is black. On the right, on the left, it's black. On the right, it's red. Sorry about that. But again, we're not seeing, we can preserve the wild type copy now, but we're losing now some inhibition of the mutation. So the next steps are probably to go to different chemistries. We'll try a different mutation. We've designed and made oligos against E815K. We'll just haven't tested them yet, getting close. But the bottom line is these require a lot of tinkering, changing the length, the chemistry, and ultimately the goal is to find some combination that will give you the ideal knockdown of the mutant allele without affecting the healthy copy very much. So this last question is, if we succeed in making a mutation or an ASO directed at a particular mutation, does that mean you need a different drug for every mutation? There's a possible another strategy, which is there may be other places in the ATP1A3 gene to target other than the specific disease-causing mutation, whether it's D801N or E815K or any number of less common mutations. There may be some variation in the gene that we can also target. And I'll explain that in a moment. So this is a picture of the ATP1A3 gene structure. And there's two elements here. There are these thick vertical bars. Those are the exons. And that's the term that refers to the portion of the gene, the DNA portion of the gene, that codes the instruction set for protein. Exons ultimately get copied into messenger RNA. The other spaces in between here are the spaces between exons are called introns. And these are segments that do not code for the protein. They do not end up in the messenger RNA, the final messenger RNA, but they help kind of hold the gene together, and there is some functional properties of the introns. Introns and other parts of the gene sometimes have genetic variation or variants within parts of the gene that are not directly related to the disease. What if we could design an ASO that targets those? Might we, and if they're common enough in the population, perhaps we could make an ASO that would work regardless of what the disease-causing mutation is. So again, I may have to explain some fundamental molecular biology to you. This is the central dogma. DNA is made into RNA, which is then read by the cell to make protein. Transiently, RNA is made with all of the exons and introns together. Makes this huge RNA that's then managed by the cell to remove the introns. That's called splicing. So RNA is made first into what's called the pre-mRNA. And then once the splicing occurs, now it's the final mRNA, which is going into the cell to make protein. So possibly the unspliced pre-mRNA can be also targeted with antisense oligonucleotides. And anything that's common in the population that may be even in the introns can be a target. And so this is where we're moving now, which is we've identified a series of common genetic variants that do not cause the disease, but are present in the gene. Some are in introns, others in regions of the gene that don't directly code for the protein. And these are now common enough in the population that if we succeeded in targeting these, we might get lucky and be able to develop an ASO that would work in a wide range of AHC cases, regardless of what the disease-causing mutation is. So in this case, this is work that was done with the For Henry Foundation that convinced the Illumina a genomics company to do whole genome sequencing on an individual with the E815K mutation and found a number of genetic variation that was on the same copy of DNA as the disease-causing mutation. If we can target these and they can be made specific to the variant, maybe we can indirectly knock down E815K. So that's one of the next phases of this. So maybe we don't need a separate ASO for every individual mutation. Perhaps we can target something common in the gene that would be carried by lots of people with AHC. However, this would require that people undergo whole genome sequencing to figure out what's present and how and what's its relationship with the disease-causing mutation. That is daily becoming easier and cheaper than it used to be. And so that shouldn't be a huge hindrance. So just to wrap up, my central question is, will ASOs work for AHC? The first question is, would eliminating the mutant allele be beneficial? We have some data now that would suggest that, yes, it might. If we can selectively remove the mutation, the mutant copy, the neurons may actually compensate and produce more of the healthy copy. We're following this study up with doing some functional studies to see if, in fact, it does, in fact, restore function. The other question was, can we actually design an ASO so it only attacks the mutant allele? This is going to be hard. It's only different. The only difference between the healthy copy and the mutant copy is one base pair. If it was multiple base pairs, it probably would be easier, but it's only one, and that makes it very challenging, but still may be possible. And our last question, which is one I just answered, which is, do we need a separate drug, separate ASO for every individual mutation? Maybe not. We're still working on that, but if we can target common variants, this may provide a more universal approach. So with that, I just want to wrap up thanking the people in the lab that have done a lot of this work. All the data I've shown you today is really the hard work of Christine Simmons, who is now a research assistant professor in the pharmacology department. She works closely with Chris Thompson, who's an electrophysiologist. I didn't show any electrophysiology tape, by the way. There's Christine, and there's Chris, and John Mark de Kaiser, who helps with molecular biology and advice. We've had a lot of help with our iPS cell technologies by a faculty member in the neurology department at Northwestern, who's Evangelos Kiskinis. And then all this work was all started back when I was at Vanderbilt in collaboration with Kevin S., who collected some of the earlier cell lines and made them into iPS cells, as well as Kathy Svoboda, who has a collection of cell lines. And of course, none of this would have been possible without funding from your generous foundations, and we look forward to continuing working with you in the future. So happy to take questions. Thank you again for being up early on a Saturday morning.
That's a very big and well thought out presentation, Dr. George. I appreciate it. If people have questions, if you don't mind putting your little hand up in the thing so that we don't all shout out at the same time. I might start with one. Just later in the presentation, you had talked about genome sequencing, and I know it is getting cheaper and more accessible. Does it help the research for us to try to push a program where we get as many sequences as we can?
It could. And along those lines, I've actually, thanks to Mary Soledino, who introduced me to some people she worked with at Illumina, I've chatted with the Illumina folks who run their rare disease program, and they are willing to sequence some of the DNAs for us that are represented by some of our iPS cell lines. I think they've promised to do 12 sequences for us for free. We're trying to get those DNAs ready to go to them as soon as possible. But I think a more holistic approach would be valuable, so that you're ready to exploit these advances.
Josh, I know I didn't raise my hand, but I'm just jumping in to say first, thanks to you guys for doing all of this work and setting this up. Thank you, Dr. George. And Simon and I have been chatting about ways to do this for the community. So maybe we could talk in Scotland to try to put our heads together, because we're definitely hoping to convince a company to do this at a really low cost for our entire community.
That'd be great. Alexander? You're on mute.
There we go. Hi, Dr. George. Looking forward to meeting you in Edinburgh in a week.
Yes.
So I just wanted to confirm your last kind of point where you're talking about like a pan mutation approach. The idea is, you find linkage between these intronic mutations and the actual mutations that are causative and pathogenic. Is that right?
That's correct. You need to know the phase, and I think that the possibilities are that you know the phase of which variant, which allele at that variant is in phase with the mutant allele.
Right. And then I guess the technical question is, are you going to use long read sequencing for this, or the Illumina platform, which uses the shorter reads, and how are you going to make sure they're both in the same phase?
It definitely should be long read technology. I've heard that Illumina is working on a long read technology. It's not commercially available yet, but they do it in-house.
Awesome.
And I think that's ultimately where that needs to go. So.
Got it. Great. Thank you.
The other way around this is, since we have iPS cells, we can always make neurons and actually determine phase by RT-PCR sequencing, which requires long reads. So.
Right. Great. Okay, thank you.
Thanks.
Julie, and then we have a couple in the chat.
Hi, I just have one question about the whole genome sequencing. If we already have that done for other reasons, are we able to provide it to whomever needs it to include what we have for the D801N mutation?
Maybe. I think we'd have to look at this by case. I think what the question Alexander just asked was, the standard whole genome sequencing is done using what's referred to as short read technology, which makes it a little bit challenging to determine the phase between the common variant and the mutant allele. That said, knowing what variants are in the whole genome is the starting point. So it is useful. There may need to be other studies done to determine if this targeting approach would be beneficial. So yes and maybe, I guess, are my answers.
Okay. May I ask one more question? And you may have answered it and I just missed it. When you showed the EK8 or the other mutation, not the D8, you'd mentioned finding similarities within there that maybe you could target a different part of that mutation, that other mutations may have commonalities. So for the D801N, is what you're saying is it could be the same? There could be within their ATP gene mutation, even though it's an E starting and my son's is a D starting, there could be another piece of information that's in there that could make this ASO treatment for all, or is it still specifically for that?
Let me clarify. So I think the mutations that cause AHC, whether it's D801N, E815K, or many others, those are extraordinarily rare in the population, right? Very rare. But there are genetic variations in ATP183 that are very common in the population. They don't cause disease, and they probably don't have any functional effect. They're just there. They mark the gene. We can find them. Those could be targeted. And if we could target those, then anybody that has those common genetic variations might benefit from that. And it doesn't matter what their disease-causing mutation is or where it is. So that's the concept. Okay. So we could target common variation also. It's kind of conceptual, but I think there's a path forward to test the idea.
All right, thank you so much.
We have a question in the chat from Cam Brecken in Canada. If knockdown of the healthy copy of ATP183 persists in future experiments, could combination of ASO with AAV be viable?
Yeah, I think this is a good concept, is if we cannot use ASOs, so if they can't be made selective for the mutation, and we are always losing some of the healthy copy, can we do something to boost the healthy copy? And certainly providing the viral vector, AAV, carrying ATP183 to the cells to supplement the ATP183 that you've lost with the ASO could be a strategy. I think the trick is that the viral vector contains the same sequence that's targeted by the ASO, or that the off-target effect may affect the virus as well. So I think we'd have to test that out. There may be other strategies as well. There are emerging technologies that some involve ASOs, some involve other things that may also allow one to boost the level of the healthy copy. And we've constantly been thinking and surveying the literature on what's happening, and there may be other ways to boost the healthy copy. So I think the concept is, yes, if we can't get selective knockdown of the mutant allele, can we just compensate for it by adding in more of the healthy copy ourselves with the AAV vector? So,
kind of destroy and rebuild kind of thing.
Yeah, sort of, if we can't do it selectively, then we can just supplement the cell. That's a nice concept. In practice, I think there's would have to be a lot of work to test that out.
Right. Toby Cohen asks, what are the next steps and key milestones?
Always a great question. I think our next steps are to continue with the D801N ASO program. The next step for that is to make some additional chemical modifications to these active ASOs to see if we can't preserve the wild type or healthy copy and get adequate knockdown of the mutant copy. I think there's a lot of chemical modifications that can be made. We're only limited by our creativity and our pocketbook. I think those are sort of the next steps, is to try to find a combination of chemistries that will work. We're also moving forward with testing ASOs that directly target the E815K mutation. The sequence is different around that mutation. It might be easier to get selective knockdown, and so we're hoping to see some selectivity there. Again, we'll go through the same pipeline to determine if it's more selective. In terms of milestones, again, I think the key milestone for us is, can we see better selectivity with the ASOs that are targeting the mutation itself? And that's a key milestone. And that's, I think, if we can't get there, or we can't collaborate with someone who has ideas on how to design them differently, that may be an obstacle. We also, another component of this is this concept of, does the cell repair itself if you knock out the mutant copy? And that's an independent line of research. And there we want to test whether or not, functionally, whether the ATP183 is restored after you've removed the mutant allele. So that's some independent work that we'll be doing over the next several months. I think, does that give you some answer?
I think so. Okay. I think I'll be interested to know, this is, in my visual brain, the way I think about it, our collaboration has been funding gene editing and AAV transgene therapy, along with this knockdown idea. So it's like 3 prongs to the genetic engineering. So I'm always wondering and looking for where they intersect. It may not be a silver bullet, kind of alluding to Cam's question, that maybe it's not one of these things, but maybe it's a combination of the 3. But
Yeah, I think that's a good way to look at it. Maybe there isn't a silver bullet. It may be that it does require a more complicated combination of therapies. Yeah, I think it's challenging because I think for FDA approval of anything, you have to prove safety. And if you have to prove safety of an antisense oligo and an AAV virus together, that might be challenging.
Right.
Not impossible, but I think it does it raises the degree of difficulty a little bit. And again, there are other emerging technologies that are coming along that I think, every month I hear about something exciting and new. They may not all work for AHC, but they're worth thinking about.
Any other questions for Dr. George? We have recorded this. We'd hope to, with your permission, Dr. George, share it probably on our YouTube channel and make it available for the other organizations to share as well. And then people might review it and have other questions. So we may send those, if you all send those through the organizations, and then we can send Dr. George the questions so he doesn't get flooded.
Yep.
I'd appreciate.
Happy to answer questions any way I can help.
Awesome. Thank you very much for your time, Dr. George, and good luck in Edinburgh.
I will look forward to seeing some of you there, and safe travels, everyone.
All right.